Welcome to our Cell Membrane Transports Crash Course by Laura and Anna, featuring Linguini. First off, we're going to talk about membrane permeability. A membrane consists of a bilayer of phospholipid molecules. It also has a barrier to water and water-soluble substances built of hydrophilic, water-loving tails and hydrophobic, water-fearing heads. Molecules such as gases can freely pass through the membrane, but other molecules such as ions, water, glucose, and urea need help getting through. Aside from gases, water has a relatively high permeability using aquaporins or water channels into the membrane. Ions are considered to have a low permeability and need proteins to travel through. Next is membrane proteins. Membrane proteins provide specificity and function to a membrane. There are two types of membrane proteins, integral and peripheral. Integral proteins are channels, pores, carriers, and enzymes. Peripheral proteins are enzymes or intracellular signal mediators. Membrane proteins are how molecules transport through the cell membrane and play a vital role in regulation. Following is carbohydrates. Types of carbohydrates are glycolipids, which make up approximately 10% of all membrane lipids. Next are glycoproteins, which is a majority of integral proteins. And then finally are proteoglycans. These chains have a negative charge which repels other negative charges. These are involved in cell-to-cell attachments and interactions, as well as playing a role in immune reactions. Finally is cholesterol. Cholesterol is present in membrane in varying amounts. Its purpose is to decrease membrane fluidity and permeability, as well as increasing membrane flexibility and stability.
Moving into diffusion, during simple diffusion, gases such as N2, O2, and CO2 will diffuse straight through the membrane with no added help. Ions will move down their concentration gradient during simple diffusion. During facilitated diffusion, the ion must match the specificity of the ion channel, whether that be the shape, size, or charge specificities. This type of diffusion will go down a concentration gradient and therefore is not ATP driven. The rate of diffusion through the membrane will be limited by the Vmax of the carrier protein, unlike in simple diffusion. Next up is osmosis. Osmosis is the process by which molecules of a solvent, water in most cases, passes through a semi-permeable membrane from a less concentrated solution into a more concentrated one, which equalizes the concentrations on each side of the membrane. Ion channels. There are both ungated and gated ion channels. In the top photo, we see a voltage-gated sodium channel. In this case, the sodium protein is made up of two negatively charged amino acids that will ultimately pull the sodium ion away from its water shell, and then the dehydrated sodium ion can diffuse through the channel. Specifics about these channels are that they never partially open or close. Instead, it's more of a all or none concept. Gated channels can be voltage or chemical sensitive and will only open if it receives a very specific signal. In the bottom photo, we see an ungated channel. This is closer to what we saw during facilitated diffusion. Instead of being sent a signal, these channels will allow an ion to diffuse based on a size, shape, charge of channel and ion. Ions simply have to meet the specificity of the channel.
Finally, we have active transports. In primary active transports, molecules are pumped against an electrochemical gradient involving a direct use of energy. The sodium-potassium ATPase is an antiporter enzyme located on the plasma membrane. It pumps 3 sodium ions out of the cell and 2 potassium ions into the cell. There always has to be a 3:2 ratio in order for ATP to be activated and the transport to be completed. This plays a critical role in regulating osmotic balance by maintaining sodium and potassium. Without it, cells can swell and burst. This transport requires about 1/5 of a typical cell's energy and about up to 2/3 of a neuron's energy. Lastly is secondary active transport. In secondary active transport, a symporter is used, meaning molecules are moving in the same direction. Unlike in primary active transport, secondary active transport uses an indirect use of energy from the energy stored in the electrochemical gradient of another molecule, which is usually sodium. Thanks for tuning in on our cell membrane transports crash course. Tune in next time.